Right now, at this exact second, your ability to remember, like, your own mother's birthday or your best friend's phone number is literally pulsing on and off like a dying light bulb.
It really is. You just don't notice it at all until you try to flip the switch at the exact wrong moment.
Right. Picture being in a crucial meeting, Someone asks you a really basic question. You absolutely know the answer. You can feel the shape of the word. You know the first letter, but your brain is just a blank wall.
Oh, it's the worst feeling.
And then 10 minutes later, you're getting coffee, thinking about nothing at all, and boom, the answer just drops into your head perfectly formed.
Yeah, it's a universal and, frankly, incredibly frustrating human experience.
Sure.
Historically, cognitive science has treated those tip-of-the-tongue moments as a glitch.
Like a software bug.
Exactly. We tend to view memory as this static database. Like, the file is either on the hard drive or it isn't. But what we are discovering now is that moment-to-moment memory retrieval is highly fluid. Your access to your own knowledge is actually governed by a biological metronome ticking deep inside your brain.
Which brings us to the core mission of today's deep dive. We are looking at this groundbreaking 2026 paper from the journal Neuron. It's by Morishita and colleagues, titled Infraslow Histaminergic Dynamics Govern Priming States to Gate Moment-to-Moment Memory Accessibility.
It's a dense title, but the findings are incredible.
Our goal today is to completely unpack the biological mechanism behind that fluctuating memory and to pinpoint the exact circuit acting as the gatekeeper to literally everything you know. And I see your visual backdrop is already leaning into this.
I thought it would help set the stage, yeah.
You've got this dynamic display of glowing neural waves syncing up with these data streams of eye movements and facial expressions.
Right, so that backdrop represents what the researchers call the integrated brain-body state. Because the central revelation of this source material is that memory doesn't exist in some cognitive vacuum.
Physical.
Highly physical. The ability to recall a fact is physically tethered to an invisible rhythmic tide rolling through your entire body.
Let's start with the source of that tide then. Because to understand why memory access flickers, we have to isolate the culprit. According to the paper, it originates in this small, ancient cluster of neurons deep in the hypothalamus.
Yep, the tuberomammillary nucleus, or the TMN.
The TMN. And the defining feature of the TMN is that it's the primary source of histamine for the whole brain.
That's right.
Okay, let me just pause you there because that immediately sounds counterintuitive to me. When you and I hear histamine, we usually think of hay fever, right?
Absolutely. Allergies.
You take an antihistamine to clear out your sinuses, and as a side effect, it makes you incredibly drowsy. So we generally associate histamine with inflammation or just, keeping us awake.
And that is the classical textbook understanding. It's essentially been viewed as a simple on-off switch for the sleep-wake cycle.
Okay.
But Morishita and their team looked closer at what these specific histamine neurons are doing during wakefulness. They used advanced imaging to monitor these cells in awake, active mice, and they discovered an entirely different, highly dynamic function.
Which is what, exactly?
These neurons exhibit what is known as an infraslow oscillation.
Meaning a rhythm that is incredibly slow compared to, standard brain waves. Because we usually think of neurons firing hundreds of times a second.
Precisely. Fast brain waves, like gamma waves, are rapid-fire pulses. They process immediate sensory input. But these histamine neurons pulse at a frequency of 0.05 to 0.1 hertz.
Wow, that is slow.
It is. In practical terms, that means they are spontaneously swelling in activity, hitting a massive peak roughly every 10 to 20 seconds, and then receding.
Okay, if I'm trying to visualize this standard conscious thought is like the choppy, chaotic white water on the surface of the ocean.
I like that.
But this infraslow histamine rhythm is a heavy, deep water swell underneath. It's this invisible tide rolling in and out, shifting the entire environment every 20 seconds.
That is a perfect way to conceptualize it. And to actually observe this tide, the researchers used something called fiber photometry.
Fiber photometry.
Yeah, they took genetically modified mice and introduced a specific protein into just the histamine-producing cells. This protein physically glows green whenever calcium enters the cell.
Which happens when a neuron fires.
Exactly. So by implanting a tiny fiber optic cable, they could measure the exact intensity of that green light. And they saw both the left and right hemispheres of the TMN glowing and fading in perfect unison on this exact 20-second cycle.
Okay, so let's do a quick recap here to consolidate this. Deep in our brains, we have this histamine clock, the TMN. It's not just keeping us awake. It is constantly, slowly surging and fading every 10 to 20 seconds.
That on.
So, to the listener, think about your own focus when you're trying to recall a tough fact. Have you ever noticed your concentration naturally ebbing and flowing on roughly a 20-second cycle? Because it's wild to think about.
It really reframes how you experience your own attention span.
But you mentioned earlier that this doesn't happen in a vacuum. It's tied to the body. How did they figure that out?
By essentially wiring the mice up to a polygraph of unprecedented detail, they built what's called a cross-validated linear encoding model.
Unpack that jargon for me.
Sure. In plain terms, it's a machine learning algorithm. It's designed to digest multiple streams of physical and neurological data simultaneously to find hidden correlations that a human would miss.
So they're tracking everything at once.
Everything. They measured the electrical brain waves, the EEG, they monitored pupil dilation, and they used deep learning software to track millimeter-scale facial movements, literally down to the twitch of a whisker.
And what they found completely challenges how we think about the brain and the body communicating.
It really does. They discovered that subtle physical movements actually predict the invisible histamine swell before it even happens.
Wait, really?
Yeah. Specifically, fast-moving gamma brainwaves and tiny nose movements consistently occurred up to 1 full second before the histamine hit its peak.
Let me push back on this, though. Are we saying a twitching nose is causing the brain to release histamine? Like, is the physical body driving the neurological state here?
It's super tempting to look for a linear domino effect, but the researchers explicitly warn against that. They use the term integrated brain-body state for a reason. The nose twitching doesn't cause the brainwave, nor is it a simple reflex.
So what's actually happening?
Think about what a nose movement represents for a mouse. It's sniffing, right? It's actively sampling the environment. The sniffing and the fast gamma waves are the physical manifestations of the entire organism booting up its processing power.
Ah, I see.
And the histamine swell is the neurological engine revving to support it.
Okay, so it's not cause and effect. They are two instruments playing the same song. The sniffing is just the visual cue that the invisible histamine wave is about to crest.
Exactly.
But couldn't this just be general arousal? Like the mouse gets startled, its heart rate jumps, it sniffs, and its brain lights up.
The data strongly refutes that, actually. If it were a general fight-or-flight arousal, you would expect an immediate spike in pupil size. Your eyes dilate when you get startled.
Right, the classic adrenaline response.
But the linear encoding model showed that pupil size actually peaks several seconds after the histamine wave. Furthermore, when the researchers later manipulated these cells directly, the pupil size didn't change at all. So this isn't a panic response. It's a highly specific, coordinated priming state.
Okay, time for another quick bullet summary. We've got a 20-second histamine wave that syncs perfectly with specific physical tells, creating this primed state. So next time you're trying to pull a memory out of thin air, pay attention to your body. What physical twitch or shift do you do right before the answer pops into your head?
It's usually something subtle, like leaning forward or a sharp intake of breath.
It's fascinating. But how do we leap from a primed physical state to proving this actually controls memory? Because seeing a correlation between a brainwave and a twitchy nose is cool, but it doesn't explain the tip of the tongue phenomenon.
Right. To bridge that gap, you have to ask a mouse if it remembers something. And to do that, the researchers used a behavioral test called trace appetitive Pavlovian conditioning.
Okay, classical conditioning. You ring a bell, give them food, and eventually they expect food when they hear the bell.
Yes, but the trace aspect is what makes it cognitively demanding. The mouse hears a specific tone for 2 seconds. Then the sound stops entirely, and there is 1 full second of total silence.
Just a gap.
Yes.
Yeah.
Only after that empty second do they receive a drop of sucrose, which is sugar water.
That silence seems crucial. The mouse can't just react to the sound in real time. It has to hold the memory of the tone in its working memory across the void and anticipate the reward.
Exactly. And the researchers measure memory expression very simply. They just watch, does the mouse start licking the delivery spout in anticipation during that silent gap before the sugar even arrives?
Oh, got it.
If they lick, they remember the rule. If they don't, the memory failed to surface.
And this is where the behavioral data mirrors our own human experience. Because even highly trained mice, like mice that have run this trial hundreds of times and absolutely know the rule, they don't lick every single time, right?
No, they don't. Their memory access fluctuates. Sometimes they anticipate perfectly, and sometimes they just sit there, totally blanking.
Just like me trying to remember my passwords.
We've all been there. And when the researchers overlaid the histamine data onto the behavioral data, a profound pattern emerged. They looked at the histamine levels just before the tone even played.
Before the cue.
Yes. During the top 10% of trials where the mice licked the most enthusiastically, their pre-cue histamine was heavily elevated. If the histamine wave was high when the tone played, memory expression was essentially guaranteed.
But again, to play devil's advocate, that's just correlation. To prove causation, you have to actually take control of the system.
And the way they did this is straight out of science fiction. They built a closed-loop experiment.
I love this part. Break this down.
It's an incredible piece of engineering. They wrote a Python script that read the calcium fluorescence in the mouse's brain in real time. The computer continuously calculated a rolling average of the histamine levels, and they programmed the system to basically hold back the test.
So it wouldn't play the tone randomly.
Right. It waited until the exact millisecond the mouse's histamine naturally swelled to the 98th percentile.
That is wild. It surfed the biological wave, it waited for the absolute peak, and only then did it play the tone.
And the results were massive. When the tone was perfectly timed to that histamine crest, anticipatory licking jumped by 39% compared to when the tone played during a low histamine trough.
Almost a 40% increase just from timing it right.
Yeah, that 39% jump proves that the physical priming state dictates cognitive success. But they wanted absolute, indisputable proof that the TMN neurons themselves were the gatekeepers. So they turned to optogenetics.
Okay, this is where we need to break down the mechanism, because optogenetics gets thrown around a lot in neuroscience podcasts and articles, but the actual process is crazy. How do you control a memory with a laser?
You start with a viral vector, which is essentially a harmless virus acting as a microscopic delivery truck. You use it to insert a light-sensitive protein into the cell membrane of only the histamine neurons.
Just those specific cells.
Exactly. One group of mice received a protein called Hailer-Hodopsin. When you shine a green laser through a fiber optic cable into the brain, this protein reacts by pumping negative chloride ions into the cell.
And what does that do?
This instantly hyperpolarizes the neuron, completely silencing it.
So they shine the green light just 1 second before the tone plays, paralyzing the histamine clock. What happens to the mouse?
Complete cognitive failure. The mice suddenly acted as if they had never learned the rule in their lives. Their anticipatory licking just plummeted. The memory was in their brain, but the gate was locked shut.
And they prove the inverse is true as well, right? A different group of mice got a different protein.
Yes, a protein called channel rhodopsin. This one responds to blue light by flooding the cell with positive sodium ions, which forces the neuron to fire.
So green light turns it off, blue light turns it on.
Right. For these mice, they made the memory test much harder. They introduced a second, fake tone that meant nothing, no sugar.
To see if they'd get confused.
Exactly. When they pulsed the blue light to artificially blast the brain with histamine right before the tones played, the mice became incredibly sharp. Their ability to successfully discriminate between the real tone and the fake tone significantly enhanced.
I have to play skeptic here again, though. If you fire a laser into a mouse's brain to stimulate it, couldn't that just make the mouse hyperactive? Like maybe the blue light just made them thirstier or startled them into licking whatever was in front of them.
That is the exact trap of behavioral science, and the authors ran a massive battery of control tests to rule it out entirely.
What kind of tests?
They proved that stimulating these neurons did not increase spontaneous licking when there was no tone. It didn't change how much sucrose they drank when it was just freely given to them. They tested their acoustic startle reflex and something called pre-pulse inhibition, which are basic neurological startle responses, and neither of those changed.
Meaning the laser wasn't just a generic wake up and eat button. It purely modulated the accessibility of the specific learned memory. The causality is totally bulletproof here.
Absolutely bulletproof.
Green light causes the tip of the tongue state, and blue light cures it, which is mind-blowing. So, brief recap. High pre-cue histamine guarantees high memory recall. By controlling these neurons with light, researchers proved this circuit directly causes the tip of the tongue state.
It's a huge leap forward.
Think about the applications for a second. Imagine if you could wear a device that perfectly timed your study flashcards to your brain's natural histamine peaks. How much faster do you think you could learn a new language?
It completely reframes education, from forcing focus to timing focus. But scientifically, this discovery forces us to follow the biological breadcrumbs even deeper.
Right, because we've established that the TMN histamine is the gatekeeper. The next logical question is, what exactly is it unlocking?
When the histamine floods out of the hypothalamus, where does it go to actually pull a memory out of storage?
The researchers tracked the signal, and they found it travels to a region called the basolateral amygdala, or the BLA.
The BLA is absolutely critical for associative learning. It's the physical structure where the brain takes an abstract sound, the tone, and biologically welds it to a highly motivating reward, the sugar.
And to understand how histamine affects this specific area, the researchers attached miniscopes to the mice.
Literally wearable, miniaturized microscopes mounted right to the skull.
That is amazing.
Using another fluorescent indicator, they could visually watch hundreds of individual neurons in the amygdala firing in real time while the mouse performed the memory task. What they were looking for is called a canonical cue-evoked population response.
Okay, that is a dense phrase. Let's translate canonical population response. Imagine the basolateral amygdala is a highly trained symphony orchestra, and the memory of the tone and sugar rule is a very specific piece of sheet music.
Okay, I'm with you.
When a memory is successfully recalled, it's not just one cell lighting up. It is a specific ensemble of neurons firing together in a perfect, repeatable pattern. That coordinated performance is the canonical response.
That's a really brilliant analogy. And the researchers used mathematical modeling, specifically amplitude scaling, to measure the accuracy of this performance. During trials where the mouse confidently remembered the rule and licked the spout, the neural firing perfectly matched the ideal canonical response.
The orchestra was playing perfectly in tune, hitting every single note.
But here's the crux of the whole paper. What happens to the orchestra when you take away the histamine? They brought back the green laser, right? They silenced the histamine clock right before the tone played.
And what happened?
The results caught on the miniscopes were staggering. In the control trials where the brain was operating normally, about 65% of the specific neurons required for that memory fired exactly as they were supposed to.
So a solid performance by the ensemble.
But when the histamine was artificially suppressed, that overlap dropped violently. Only about 9% of the correct neurons managed to fire. The canonical pattern completely dissolved.
From 65% down to 9% accuracy. That is catastrophic. But here's the detail I find the most fascinating. The overall electrical activity in the amygdala didn't drop.
Precisely. It wasn't that the amygdala went to sleep or shut down. The neurons were still firing, but they were firing chaotically. They completely lost their coordination.
To bring it back to the orchestra without the histamine swell, the musicians are still sitting on stage and they are still playing their instruments, but they are totally disorganized. The violins are off tempo. The brass section is playing the wrong song entirely.
It's just noise.
Right. So the histamine isn't a musician, it's the conductor. Every 20 seconds, the conductor sharply taps the baton on the music stand, calling the entire room to absolute focused attention. That is the priming state.
Yes.
When the tone plays during that focused window, the orchestra perfectly belts out the memory. But no histamine. You get a noisy, disorganized mess, and that mess is the tip of the tongue state. The memory is physically there, the cells are active, but they just cannot organize the signal.
And the researchers cemented this by implanting optical fibers straight into the amygdala itself, hovering right over the terminal endings of the histamine neurons.
Oh, wow.
By just shining blue light directly onto those specific terminals, basically activating the conductor right at the podium, they enhanced the mice's memory discrimination. The circuit is verified all the way from the clock to the conductor to the performance.
It is so beautifully mechanistic. So let's do one last recap and pull all these threads together. We have an infraslow metronome in the deep brain ticking every 20 seconds.
Yep.
It syncs with our body's physical exploration, like sniffing or shifting focus. When it surges, it floods the amygdala, organizing chaotic neural firing into a sharp, clear memory. This completely shatters the idea that a memory is just a static file you open.
It is a dynamic, biologically dependent performance that has to be flawlessly timed.
Which forces us to ask a pretty profound question about the nature of disease. With this precise circuit now mapped, how might medical science eventually treat severe pathologies of memory?
It opens up so many doors. We're talking about conditions where memories become trapped behind a locked gate, like in early Alzheimer's or Lewy body dementia.
Or even the inverse, right? Conditions where the gate is broken open and memories are violently, uncontrollably intrusive, like in severe PTSD.
Exactly. If we can target and stabilize this histamine metronome, could we theoretically rewrite how a patient accesses their own past?
The clinical implications are just staggering. But stepping back from pathology for a second, the final question the source material leaves me with is a philosophical one about our own healthy biology.
What's that?
Why on earth are we built this way? If it is so crucial for survival to remember where food is or who our friends are, why isn't the memory file just permanently open? Why isn't the conductor just standing at attention 24/7?
Because the brain is bound by the brutal economics of evolutionary biology. The source material points to one overriding reason for this fluctuating state, and that is energy conservation.
Power-saving mode.
Literally.
Yeah.
Maintaining sustained neural activation, keeping the brain in a state of constant, perfect memory readiness demands massive amounts of ATP, the cellular energy currency. The brain is already a huge metabolic drain on the body. Perfect recall is simply too expensive to run continuously.
It's like leaving every single high-powered application running on your laptop at once. The battery would just drain in an hour. So running the histamine conductor on a 20-second low-duty cycle saves power.
Exactly. It manages the biological cost while maintaining a crucial balance between exploration and exploitation.
Well, so?
Well, when the histamine wave is at its trough, the brain is in a lower power state, untethered from rigid past associations. That makes it highly adaptable to exploring novel sensory input.
Taking in new things.
Right. And when the wave crests, the brain perfectly exploits its stored, learned knowledge to make a critical decision. It's a highly sophisticated evolutionary power-saving mode.
I really love that framing. It turns what feels like a frustrating flaw into an incredible feature. So, to everyone listening, the next time you find yourself staring blankly in a meeting, desperately trying to summon a name or a statistic that is right on the tip of your tongue, take a breath.
Don't panic.
Right. It is not a sign of cognitive decline, and it's not a glitch in your hardware. Your brain is simply operating exactly as millions of years of evolution designed it to, resting in an elegant energy-saving trough.
You literally just have to wait a few seconds for the tide to come back in.
So pay attention to your own rhythms. The next time the answer finally pops into your head, notice if you just shifted your posture or took a sharp breath. You might just be feeling the crest of your own histamine wave. Keep questioning your biology, and we will catch you on the next deep dive.